The subject of my talk is about my previous research, but I'm happy to talk about sort of my current work at the end of this. So the research I'll be presenting on was most of the content of my master's thesis at the University of Massachusetts, which is in Amherst, Massachusetts. I worked with Dr. Chris Sutherland, who is now at the University of St. Andrews. Chris is a fantastic advisor. I'm also a great ecologist and statistician. I've changed the title to be a bit more descriptive today. So that title reads Optimization and Integration for Improved Ecological Inference Using Spatial Capture-Recapture. So at the heart of my talk are these two ideas. First, optimization, which is optimization of spatial sampling designs, or in other words, like where to put camera traps. And second, integration in terms of bringing together different types of data, which in this case means camera trap data and GPS telemetry data. So just to set the scene here, understanding the causes and consequences of changes in animal populations is fundamental to ecological questions. But to investigate such questions and avoid spurious conclusions, we need to have good estimates of population size. This motivates the need for the development of rigorous methods to monitor populations. The snow leopard is a great example of this. It's a secretive species, and they're also of serious conservation concern, with only around 3,000 individuals remaining. But we need better estimates to help guide conservation efforts. So getting kind of right to it here, spatial capture-recapture, or what I call SCR, is the leading method for rigorous estimation of population density. The basic SCR model is built on an implied model of how individuals use space and a model of how those individuals are distributed in space. So it was conceptualized in the early to mid 2000s. The model is still relatively new, with lots of opportunity for improvement. SCR data, though, can be challenging to collect in large quantities, so we need to have new ways to provide data to the model to obtain better estimates of density and other parameters of interest. So today I'll focus on two advancements in this regard, which I mentioned with the focus of my master's work, sampling design and data integration. So I'm just going to kind of start off the bat with a little mini lesson on spatial capture-recapture, for those of you who might not be familiar. So let's say we want to sample a snow leopard population to estimate snow leopard density. Snow leopards are a good candidate for SCR because they're individually identifiable, because they have spots all over them that we can identify uniquely. Individuals have home ranges, which is the local area they use to find food or reproduce, and you can think of this as related to territory. You can see this animal kind of moving around there like that. To understand how many snow leopards there are, it's helpful to know how much space space each individual uses, and we can quantify this area using a simple but special model. And this is kind of what that model looks like. It describes the probability, P, of an individual using any location in space. So you can kind of see it's moving around somewhat accordingly. In the basic SCR model, for now, we can say space use can be used interchangeably with detection, and this means an animal is more likely to be observed in locations where it's more likely to use. The model is symmetrical, and we can look at it in just one direction. We see that the probability of the individual using a location in space decreases from the center of its home range. We call that center point the activity center, and that's denoted as S sub I, as you can see here. That's really just used for statistical convenience. It doesn't need to have a biological meaning. For more detail, we can imagine that we're viewing this model from above right now, and we can take that slice of it and view it from the side like a piece of cake. And this is the same function. I'm sure it's familiar to many of you. It's the heart of the SCR model. We'll see this frequently in the coming slides. Detection probability, P, which is on the vertical y-axis, decreases along the horizontal x-axis away from the activity center, which is at x equals 0. You notice that this is a bell curve that has been cut in half. We call it the half normal model. There are two parameters that work together to change the shape of the curve. The first is P naught, which describes the detection probability at the activity center. So that's at the center of the animal's home range. And then sigma, which describes the spatial scale of detection. So it's sort of a distance measurement related to the radius of the animal's home range. So we estimate these parameters from the data, meaning we collect our data, then we can guess which parameter values are most likely to give rise to those data. These parameters are P naught, which I have there. That describes baseline detection. Sigma describes the space use for the spatial scale of detection. And then we also estimate density, which is really the main objective, and it's the second part of the model. The idea here is that if we can estimate our probability of detecting an individual, and we can also detect some individuals, then we can estimate the population size. So here we have big N equals little n over P. And that's the number of individuals detected divided by the probability of detecting an individual. So basically, if we see 6 animals and we know there's a 50% chance of detecting any individual, then we can estimate that we have 12 individuals. So that's really at the heart of it, but SCR improves on this idea by making detection probability spatially explicit, as we already saw. So let's talk about the data. We can imagine our study area is this presentation slide, and we know there are some snow leopards out there moving around their home ranges. In reality, we don't know how many snow leopards there are, but to find out, we can deploy camera traps, which are devices that take photos of individuals so that we can identify them. Just gonna let that run through there. So here's those camera traps. We can keep track of which individuals are detected on which traps. We can see the traps are numbered. This simulation that I'll go through is just one sampling occasion. We can say an individual is detected if it overlaps with the camera. So you can kind of watch one of these snow leopards and see where it's detected, and then we'll see how the data is collected afterward. So they're moving around. Snow leopard B is detected on trap 3, snow leopard A is detected on trap 4 and trap 2, and then we can organize this detection information. We can make a table where each row is a snow leopard, each column is a trap. Corresponding value in the table is a 0 if the individual isn't detected, or 1 if it is detected. So we have a snow leopard A here is detected on cameras 2 and 4. So it gets ones there, and then the zeros for cameras 1 and 3. This is the recapture part of spatial capture-recapture. It was detected or captured on trap 2, and then recaptured on trap 4. So recaptures help us estimate P0, which is the parameter I talked about earlier. And then further recaptures at separate traps, like this one here, also help us estimate sigma, which is the scale of the home range. So snow leopard B is detected on camera 3, increasing our sample size. Snow leopard C is too far away. It isn't detected on any of the cameras. So in reality, we wouldn't even see that individual in our capture history. We can start to get an idea for territory size from the pattern of these encounters relative to the distance between traps. Territories are at least as large enough to have detected individual A on cameras 2 and 4, but not much bigger since individual B was only detected on camera 3. So we can call this table Y, and Y is indexed by rows and columns, and that's I and J. Each individual gets an I for its row, and each camera gets a J for its column. Now that we've formalized our data collection, we can describe the system with a statistical model. This is the most basic SCR model that I'll go through. So individuals, represented by their activity centers, are distributed uniformly or randomly across the study area, which is the big S. The observations in the table that we just saw, Y, are dependent on the activity center locations and on the result of a Bernoulli trial with probability P. So of course, a Bernoulli trial is like a coin flip, but instead of a 50% chance of getting heads, this coin is weighted, and it's weighted by the probability P. P is the detection probability, which decreases with distance from that individual's activity center, as described by P0 and sigma. So kind of to summarize here and bring it all together, SCR is a model of individual space use related to detection. Space use provides a spatially explicit representation of the area being sampled. That area defines detection probability, and hence provides objective or unbiased estimates of N, which is abundance, or D, density. But in order to estimate the spatial scale of detection, we have to estimate sigma. So as we saw in our Tor example, to estimate sigma, we must detect individuals at multiple locations, which are also known as spatial recaptures. You might notice that this description is leading us a bit, and you might be putting together that the quality of our estimates depends on where we sample. So really, this comes to a sampling design problem. And that idea has been the focus of, or was the focus of my first chapter of my master's thesis, and it's on optimal sampling design for SCR. This was a collaboration with my previous advisor, Chris Sutherland, who I mentioned, along with Ali Nawa from the Snow Leopard community and Andy Royal from USGS. So why focus on sampling design? Well, so we can answer a different question, which is how well will we be able to estimate what we care about? In an often case, that's density. Or in other words, will we be able to get a good estimate? And if the answer is yes, well, the answer is yes. We have an expectation of the data we're going to collect. We can build that expectation by having good guesses for P0 and sigma from the literature or from pilot studies. That gives us an idea for home range size, which we can use to figure out how far or close our camera traps need to be. In statistics, the quality of our estimate is mostly a function of data quality, of course, and we have a good sense for SCR data from our mini example. As we saw, there are 3 components of SCR data. You recognize these now. They are the number of traps, the number of recaptures, and the spatial pattern of encounter histories or spatial recaptures. So this means that there's a trade-off among these elements. We want to capture a lot of individuals, but we also want to have a large number of spatial recaptures where individuals are captured on more than one trap. But these objectives are competing. We can't do both. We need something in the middle, something that's kind of just right, which is why I like to refer to this as the Goldilocks paradigm of design. So we're going to go through 2 examples here. These are both the same area. The light blue is the area we're sampling, and then the same number of traps, which are the crosses. And and the same number and placement of individual home ranges. Those are the red circles there. So on the left, the traps are spread out. We detect a lot of individuals, but only on a few traps for each individual. And then on the right, we only capture a few individuals, but we detect those individuals on a lot of traps. So that's really the basic idea. Previous research has been helpful in providing guidance on how to manually create designs within a rectangular grid like this, but there aren't any formal approaches for sampling design in irregularly shaped study areas, which are far more common than rectangles. One of those regions that motivates our research is this one here. It's the sampling area in red, which is in the Astor region in Pakistan. The country is white on the right. And we're sampling for snow leopards on the western edge of the snow leopard range, which is in yellow. The study area shape is irregular, and further, it has snow-capped mountains and river valleys, as well as military bases, which means a lot of areas cannot be sampled. So after accounting for that, we're left with the resulting possible sampling locations, which are on the right here. But let's say we want to sample this area. Even knowing how big a home range is, what would you do? Following previous recommendations, it's impossible to know the best design since we can't use a rectangular grid like I showed in the previous slide. So to solve this problem of optimal design in irregular areas, we present a new approach. It has two steps, and the first is to identify a sensible design criterion, which is a criterion or a measure of how good a design is. That criterion tells us the expected information content of a design based on our understanding of home range size and detection. We can formally optimize that criterion over thousands of possible designs using an algorithm, which I'll show in the next slide here. So identifying a criterion, our framework is flexible in that it can utilize any criterion based on the SCR model composed of P naught, sigma, and density. We propose 3 criteria that explicitly leverage the encounter process. These follow from what we talked about earlier, which are captures and spatial recaptures. So criterion 1, we'll call the space filling criterion. It maximizes the probability of detecting any hypothetical individual at least once. So it's really about captures. Criterion 2 is the space constricting criterion. It maximizes the probability of detecting an individual at more than one trap. So it's about spatial recaptures. And then number 3 is a hybrid approach. It simply combines the 2 previous criteria in a pretty straightforward way. So now we're going to look at how we derive these criteria, which are these measures of data quality. So the basic idea brings us back to this curve. So using the detection function, we can plug in x values, which are distances from hypothetical activity centers, and then we can get back y values, or detection probabilities. We can do that many times and organize these results in meaningful ways to tell us how likely we are to detect any individual in a set of traps. So rather than creating and evaluating designs manually, we can use an algorithm to do this for us. It can do a lot more in a lot less time. So what we use is a genetic algorithm, which is one of the first uses of this specific algorithm in the field of ecology. We packaged this in an R function, which is called SCR-design-GA. I'll show that later. It's in the Oscar package, which is for open source SCR. So let's say we have 50 traps. It starts by generating a population of designs. So let's say we have 100 designs, and those are made totally randomly. We can then score those 100 designs using the criterion that we just talked about, and it selects the top designs. So let's say it selects the top 10% highest rated designs. It then breeds those 10 designs, swapping pieces of them among each other to generate 100 new designs. The process then repeats for as long as you want. We recommend doing this about 1,500 times for this part of it. And then I'll just show one more here. So it scores and selects the designs, it breeds new ones. And then 1,500 iterations later, it terminates and returns the best design. So we can see what this looks like in action. So this is for the space filling criterion. The gray area is what we're sampling. The small dots are the possible trapping locations to select from. The bluish larger dots are the traps that are selected. So what you'll see here is that the design starts off totally random, and then through time, the pattern of the best design emerges, which in this case is the traps filling the space evenly. So this is what that looks like. You can see that the pattern kind of starts to emerge as traps sort of start spacing themselves out more evenly. And that's the final design. So we have a function in the R package Oscar, which we can use to generate designs. It takes 3 things. The first is the study area components. Second is the number of the traps. And then 3 is the parameter of the parameters of the species and the related design criterion. We can see what these designs look like just from a quick example here, going back to a regular area. So the space filling criterion fills the space evenly. It should detect a lot of individuals. The space constricting criterion samples a smaller area. It should get a lot of spatial recaptures. And then the hybrid approach does a little bit of both. So it spreads out like a space filling criterion, but it has some clusters of traps like a space constricting criterion. So you can see some traps are really clustered together closely, but others are farther apart. The criterion tells us how well the design performs relative to a single objective. So to get a more comprehensive understanding, we use simulation to evaluate design performance, and I'm going to demonstrate that here. So we start with our study area and our design. In this case, each of the 3 designs. We simulate some individuals. We generate capture data, as we saw before. It's the table that we called Y. We then recover estimates of density using SCR, and then we measure performance by seeing the difference between the simulated values and the estimates. And that's kind of the beauty of simulation here. We generate the true value ahead of time, so we know how well that we did. In most cases, as I'm sure you know, this measure is called bias, and we want to minimize it. So we want no bias. We want a bias of 0, which you'll see in the results here. So bias is on the vertical axis. A bias of 0 is shown by the horizontal black line with reasonable bias shaded around it in gray. So we made designs with 3 levels of traps, or 3 levels of numbers of traps, which is 144 traps, 100, and 49. So we expect worse performance as the number of traps decreases. The 3 designs are the space filling, which is in the fuchsia color, space constricting in blue, and the hybrid design in purple. The confidence intervals here just show 50% of the core estimates to make things a bit easier to see. We can see optimized designs perform very well in general. And then with few traps, criterion 2 remains unbiased, which is really great to see, especially because criteria 1 and criteria 3 struggle at low effort, which is due to extended trap spacing in sparse arrays. Not shown here, but precision and accuracy also follow these trends. So some take-home points. Where to sample depends on the SCR model, and that's informed by the species ecology. Previous recommendations aren't very helpful in this regard in irregular realistic sampling scenarios. So the SCR design GA framework produces a greater amount of expected information, leading to more accurate estimates. The framework is flexible. It can be used for any species in any study area. It can also be used with new criteria. All right, and then some of those alternative criteria could be something that account for spatially varying density. Ian Durbach had a paper about that in 2021. Or multi-species sampling, which is something we've been working on more recently. Basically, you can split up traps and then have some traps go towards one species and some go towards another species. Another sort of future direction is sampling with multiple data types, which are sparse cameras where you'd be using those to detect individuals and then using telemetry to estimate sigma. That's separate from what I'm going to talk about next, but sort of a really related idea. For more information, you can find some of this on my website, which is gatesdupont.com. Materials are linked on the homepage, which is right there. We also have these materials in several forms, vignettes and help files, a recorded workshop lecture, and the paper that we published in Ecology. Okay, quick breather. That's the end of chapter 1. I'm going to get a little drink here. All right. So chapter 2 is on telemetry-informed cost functions in SCR. This is a collaboration with Dan Linden from NOAA, and again with Chris from St. Andrews. This chapter is on integrating data from things like GPS collars into the SCR model, and our study species here is the American black bear. So cost functions are about landscape resistance, which is about animal movement. Movement is the primary mechanism of response to environmental conditions, such as moving to cooler or warmer areas, or interspecific interactions, such as predators moving towards prey. Fundamentally, though, movement is structured by the landscape, and this concept is termed landscape resistance. Resistance is really about a reconsideration of distance, or thinking instead about functional distance. So, for example, if you want to cross a river, it can be faster to take a bridge downstream rather than swim straight across the river, even though the swimming path is shorter. This is really important. So, for example, a paper published in Science showed that we can detect how landscape structures movement even across a global scale. And here they showed that areas with a greater human footprint were home to mammals that moved around less. And that's likely because they had less available space. So what we want to know is how this works at a fairly fine scale. There are 2 methods to estimate the effective landscape resistance to movement. Previous methods have mostly been imprecise or not informed by relevant data, instead relying on expert opinion. Simultaneously, though, data integration has been growing rapidly in popularity. One especially complementary method is the integration of telemetry data with SCR. We saw that SCR methods collect just a little data on a lot of individuals. Excitingly, telemetry data collects a lot of data, but only on a few individuals. There's no method, though, that combines data integration with estimation of landscape resistance. So here's our setup. We can use camera traps to get a little information on a lot of individuals, as I said, and then telemetry data or GPS data to get a lot of information on just a few individuals. But we have to model these data types mostly separately. We can combine them by estimating a parameter that they have in common, and then that parameter is called cost or landscape resistance. So we'll see what that looks like. So if we have a road and an individual's activity center sits right next to the road, that's kind of what it would look like. The road is incurring a cost on the individual's home range, so the bear doesn't want to cross the road, but we can remove the road to see what it's doing to the home range. So the home range is no longer symmetrical, as we saw in one of the earlier slides. It now follows a contour on the road. We can see how this works by comparing paths to two different pixels that have the same probability of space use. The path to the right includes the cost of the road, so it's shorter, whereas the path to the left does not have to go through a road, so it can be longer. We use cost as a way of defining an equivalency between distances like this. So we call this reconsideration, I don't know why I keep saying that, ecological distance. Here's how we include it in the model. We have the detection function, which uses distance, and we swap in ecological distance. What's not shown is what's under the hood. It's our calculation of ecological distance. That's what's known as the cost function. It's just 3 lines of code in R. It's carried out by the package gdistance. Conceptually, we estimate the value of a parameter that allows us to bend space in a way that makes our data the most likely. So that's our model for the SCR data with ecological distance. But what about the telemetry data? For that, we use a movement model. So in our movement model, we want to understand where an individual is going to move after each consecutive step. We start with a track like this. The individual has an activity center S, and the individual is now to position U. So we want to know where it's going to go next. We have the home range model to describe probabilities of space use in the home range, and we can use that same model, but shrink it to the size of each step and center it on the individual's current location. The probability of where the animal moves next can be calculated by adding these two models together. So we can look at this a different way. Where the individual moves next is a function of its distance from its current location, plus the distance from the activity center. We can translate that into this formula. The important thing to note is that we can also include ecological distance in this formula. So we can use ecological distance in both the SCR model and the movement model. And the distance is estimated from a single cost function, which estimates the effect of landscape resistance. So just as we did in my first chapter, we'll use simulation to evaluate performance of this method. So we generate a cost surface, and that's something that looks like this. The camera traps are again crosses. Individuals have activity centers in the landscape. The black box is an area that we'll zoom in on to see one individual. We generate a movement track, which is the purple track, within the home range, and then we record that track. We can collect these same data in our camera traps so that every time the track overlaps with a camera trap, we record that as a detection at some standard probability. So the dark circles are where the animal is detected, and that inset number shows the number of captures on that trap. And now we have SCR data, which is what you can see in the top right corner there. We wanted to see how our new model compared to other models. So we compared performance among 4 models. We used simulation, and we also used a case study so we could try out our model on real data. The case study data were collected on black bears in New York State. We used the non-integrated standard SCR model using Euclidean distance, which is distance as the crow flies. Then the non-integrated SCR model using ecological distance, the telemetry integrated model with Euclidean distance, and then our full model, the telemetry integrated model with ecological distance. So we can check out our results by using these models. We compared the non-integrated, which is in red, and the integrated model in blue, which includes ecological distance. We can evaluate bias. The main plot is for our cost estimate, and the inset plot is for our density estimate. The inset number is the number of collared individuals that we include. So integrating telemetry data improved estimation of cost. SCR data alone was not able to recover an unbiased estimate of cost, but it's important to consider the simulation design here. Adding data from more individuals showed little improvement since the model was already doing very well. And then finally, estimation of density remained unbiased. We know we can reliably estimate both parameters at the same time. This is the case study from the New York black bears. So the green spatial covariate is percent forest. The traps are again crosses, and then we have 3 bears that have telemetry data on them. So we saw similar results from this case study. Integrating telemetry data improved estimation of cost. You can see there are smaller confidence intervals here. SCR data alone was not able to recover a significant estimate of cost. Adding more individuals showed that variation among individuals should be considered. And again, density estimation remained consistent. So some closing points on chapter 2. First, data integration is a promising approach, especially when the process you want to understand is fine scale, as is the case with landscape resistance. Integrating telemetry data via movement model was a perfect fit in this case, providing more information by using an explicit model of the movement process. Future research could focus on using a more direct treatment of multi-scale selection or selection in the home range versus within the movement path. Some methods are already out there and could be easily included in our framework. And yeah, so a bit more on these future directions. We could allow variation in fixed rate or more direct treatment of multi-scale selection. And then exploration of spatially varying density together with landscape resistance from a single surface. So you could have the same surface that kind of determines how density is patterned in the landscape, and then also how movement is patterned in the landscape. Ecologically, examining resistance within home ranges versus during dispersal events could also be interesting, sort of a multi-scale connectivity idea. So finally, this chapter is more recent, so we're working on getting more materials out there. Right now, we have a manuscript, and we'll be including this model in OSCAR and provide some documentation for it. That's the end of chapter 2. Just a couple more general discussion points. 2 broad takeaways. These are technical details that I came to realize are really important, but at the same time can be overlooked pretty easily. The first one is that if you want your new method to be widely adopted, try to make it familiar. If you step back for a moment, you'll realize that we spend a lot of time going back to that detection function. In chapter 1, we used it to calculate design criteria, and in chapter 2, we used it to describe step selection in the movement model. It came from the heart of the SCR model, which is used to describe the home range. So when we saw it later, it was familiar, we could understand it quickly and start using it without having to read an extra book. Second, a lot of thought goes into the simulation design for methods studies like this, but it's worth giving them a second thought. So in brief, to evaluate new statistical methods, simulation studies can be a bit circular. Typically, the model that we use to simulate test data is the same as the model we use for estimation. This can be fine in some cases, but in other cases, particularly with more sophisticated estimation models, the circularity can create blind spots. So in the design chapter, the evaluations were simple and the circular approach was adequate. But in the data integration chapter, parameterizing the model was a bit tricky and presented problems that we would have missed if our data generating model had been circular and self-fulfilling. So I wasn't able to talk about that in detail, but I can use this sort of as a plug to read the paper. Certainly some good food for thought. Finally, just wanted to say that being a researcher from the United States who's never traveled to Central Asia, my knowledge of snow leopard biology from the first chapter will obviously pale in comparison to the knowledge of local communities. The success of this project was mostly in part due to having the honor of working with those people who live in the local area and have the local knowledge, showing the challenges faced in those areas and highlighting the challenge of designing population monitoring programs to estimate density and define future conservation actions. So we made all of our design work open access and open source as much as possible. So far, it's being used in Pakistan and Mongolia for snow leopards, as well as South Africa for common leopards. So again, my knowledge of these predators cannot compare to that of the communities with intimate ecological knowledge from living among them, but I know that by complementing the research efforts of local conservationists, we can collaborate in a way that allows us both to learn more about these important species than had we not entered into such a dialogue from the start. So with that, I'm just going to say thanks to a few people who I mentioned along the way: Chris Sutherland, Ali Nawa, Andy Royal, Dan Linden, And then I got funding from UMass OEB, Panthera, through the Savin Snow Leopard Grant, and the Snow Leopard Foundation, and also the Snow Leopard Trust. And that's all. So any questions, I'm happy to take them. I just hop back